And to have a part that got made, you would need to attach a promoter and a ribosome binding site, and then the actual part that gets coded.
I mean, I know you're saying if these things don't work, is there a way to, would you be messing with the sequencing, or how would you try to make these basic parts do what you thought they should do?
Well, the issue is not necessarily that the part would be doing something completely different from what we expect, but different combinations of parts may make, one combination of parts may make a lot of protein, another combination of parts may make a lot less protein. Sometimes you want a lot of protein, but even if you want lots of protein, you have to be careful. If you make too much protein, then the cell actually falls behind. The cell doesn't just sort of synthesize the protein and then stop there. Then there's cellular machinery that's devoted to folding it and moving it around. And if you make so much stuff that the cell becomes overwhelmed, it can become toxic. And it can, you could imagine a case where I have two options. One makes 10 times as much protein as another, and I would want to use the one that made 10 times less protein because the cell can keep up with that level of protein. It doesn't cause the cell to die. And how toxic a particular protein will be under what circumstances, especially when you're combining a bunch of stuff together, is something that you usually have to check. You can have guesses. You can know from previous experience or from papers in the literature that this protein might be a difficult protein to work with. And in that case, it usually requires making a couple of parts, some of which might make more, some of which might make less, and seeing which ones are optimal for your system.
So, coming at this from a chemical engineering viewpoint or pathway, what does it feel like working with like biological systems versus chemical systems?
One of the great advantages of a degree in chemical engineering is I don't know any chemistry. It's actually, all of the chemicals that I dealt with were A plus B going back and forth to C, which goes to D. It's usually, we look at it from a fairly mathematical approach, and we're looking at types of reactions instead of very specific reactions. So when someone said, well, we're going to analyze how fast this reaction happens, but I can look at the biological molecules, but I can also just sort of make them wave away in my head and replace them with A and B and C and D. So it's the basic training there is readily applicable.
Is it as reliable though, working with biological systems versus chemical systems? Because aren't there many more unknowns involved?
Yes. It depends. There are definitely lots and lots of interactions, many of which are not known in a biological system because there are so many different molecules there. If you look into things like combustion, there's actually a lot of molecules popping up there. I mean, it's orders of magnitude less, but there are mathematical techniques that are good at dealing with systems like that. One of the issues is that if you have 1,000 different molecules, for example, and let's say one of those molecules can have interaction with another 10 or 20 or 30 of the other 1,000, then you have to characterize how or how quickly each of those interactions becomes, and the sort of amount of data that you need to know to really pin things down is gigantic. And it's also very hard to get because the cell is, it's very difficult to say, well, okay, I've got a cell with thousands of reactions occurring simultaneously. I only want to look at one. Well, they're all interconnected, so it's very difficult to tease out a single reaction. I always forget who said this, but someone once said that the way that you get data in molecular biology is that you freeze the cell in a certain state and you take a look at what's happening with one molecule or a couple of molecules, which is like trying to understand the game of football by taking random snapshots of the field and trying to figure out the rules. So that said, there are things that you can do, and in fact there are people that are specifically very interested in how do you deal with systems where the euphemistic term is they're data poor, where you don't know a lot of the actual numbers. And there are tricks that you can do. There are analyses that you can look at to say, well, you know what? We don't know any of these numbers for all these reactions over here, but we've done a test, and there are various kinds of tests. One of the simplest ones is I just plug in a bunch of numbers. I have no idea what they are, so I just try a bunch of different numbers. And I find out that the numbers over here aren't really that important. There's a big reaction network here. These numbers are important. These numbers are important for my given output, whatever that is, like activation of this or growth or whatever. But these aren't. So it kind of, there are techniques that can point you towards the most important parts of the system when you have a big complex system and you don't want to measure every single little thing that's happening in it.
Oh, excellent. Okay, so one question I had, I don't know how well you can answer this, but do you know what sorts of basic knowledge the students involved in the IGEM project are required to have?
I think you definitely need a basic undergraduate level understanding of molecular biology and genetics. It definitely helps to have some lab experience. There's pretty much any time you're working in molecular biology, you are going to be pipetting small volumes of liquid from one tube to another a lot. And it also helps, it also, let's see, this is gonna be a long drawn out answer on tape. So I'll start over. Students in IGEM should start out with basic undergraduate level knowledge of genetics, molecular biology, and I think that it's critical to have a strong math background, either applied mathematics or calculus plus differential equations.
So for like the high schoolers, for example, are they as involved as the undergraduates in the actual project, or?
I think the high school students are, so the high school students, let me think about this again. I think the only part where the high school students are less involved is in the research direction, because they necessarily have less experience with what is possible. So but the high school students are, within the project, and the high school students started after the project had began. So when we were talking about project design, we were talking about, very grandiose terms, the kinds of things that we wanted to do. And I think it's hard, unless you've had several years of undergrad, to be able to speculate very well. simply because it's difficult to tell, what's possible and what's